perfect. So to continue on Lloyd's topic, I will be covering what a small sequence of nucleotides is sufficient for a virus to go on through its life. So I am Professor Aurelie Rakotondrafara. If you're still wondering where this name is coming from, I'm originally from Madagascar, and yes, I do speak French. So the title of my talk today will be to present you a plant virus race, a sequence, spatial, temporal sequence of viral infection. So viruses have been have been around for centuries, and as a substance that is contagious, spreading diseases. And the earliest account record of viral infection was from the was from the 14th century BC, where you can see this ancient Egyptian stealth tomb but they picked polio-affected priest. You can see here this foot drop deformity that is indicative of residual paralysis from polioviruses. The historical, first historical record, it was from the 1600s, from the smallpox outbreak in the Aztec Empire, where they were describing 200,000 people died of rashes and pustules all over the body. This outbreak of disease played a key role in the ability of the Spaniard to conquest the Aztec Empire. What is key is that it was thanks to an agricultural catastrophe that plant viruses have been discovered, and all started in tobacco. So it was in the 1800s where tobacco was a cash crop. It was all about money. I can tell you that by that time, 5 pounds of dried leaves of tobacco was equivalent to 3 times the value of the same amount of silver, to telling you that it was money. And what happened in 1879 in the Netherlands, the tobacco farms were swept by a disease that left the plant stunted, and with this particular discoloration of the leaves all over it. The Dutch people begged biologists to help them to understand what was happening, but at the same time, what should they do about it? And it took 50 years of research that in 1935 that they discovered, thanks to the development of electron microscopy, how a virus really looked like. And here, you can see here the particle of a virus, and it was tobacco mosaic virus. So one of the characteristics of these viruses are they are among the smallest microbes. They cannot be seen under a light microscopy. What you can see here as an example, the size of the plant or animal cells in the 10 to the minus 4, 10 to the minus 5 millimeter, and here we're talking about 110,000 matches smaller. And the question is, what are viruses? So to give you even a better appreciation of how cool are viruses, by the way, I'm working on viruses, that's why I find them cool. But human, for us, each of us, we are made of more than 50,000 trillion of cells. And each cell comes in different shapes, they have different function, and inside of each of the cells, you have about more than 20,000 genes. And as Lloyd just mentioned to you, if we line up the DNA that contains the 20,000 gene coming out from one cell, it will be indeed 6 feet long. Such a complexity is to contrast with viruses. What are viruses? Viruses are just made of protein and nucleic acid, RNA or DNA. The protein, in fact, is just like a shell, a protective component that cover the genome of a virus. When I'm talking about how simple and small are viruses, if we look at the genome size of the tobacco mosaic virus, we are talking here about 6,000 nucleotides. Remember what Lloyd told you. Chromosome 1 was 249 million nucleotides. And here, we are talking only about 6,000 nucleotides for a virus. The number of genes, 4. The smallest known viruses, it's another plant viruses, tobacco necrosis viruses, and in this case, it just needs 1 gene. 1 gene sufficient to create a major catastrophe in agriculture, as indeed plant viruses are known as major pests where they're able to decrease not only the yield, but also like to decrease the quality of the produce. As you can see here, different symptoms of deformity, also like necrosis and yellowing and etc. on the leaves. But like my question is, but how do they manage to do that? How do viruses cause disease? You know maybe about bacteria, like fungi, but viruses like are completely different. They do not eat the cell, this is one thing. They do not produce toxin or digestive enzyme that will like chew up the cell wall of your crop or something. But what they do, simple as that, they just take over the cellular machinery and the space, and that is sufficient to disrupt the cell function. And indeed, by taking over the cell machinery, you just disrupt, you just disrupt the normal function of the cell. So viruses, indeed, what they're doing, they are programmed to go on through their life cycle. They are programmed to turn on the cells literally into a virus-making factory. The idea of just making copies and copies and copies of their viral genome. How do they do that? They take over the cell enzyme, the molecule energy inside their host to be able to make copy of the genes, the genomes, and make copy of those proteins. And then what is cool about viruses, they're able to self-assemble themselves. They take those copy of nucleic acid molecule, those copies of the protein, and they're able to make new viruses. And it's as simple as that, how viral life cycles goes on. So like today, what I wanted to show you is about this spatial and temporal like sequence of events in a plant viral infection. Something to understand the virus as a goal. Its goal is to enter the cell, then once inside, to make copies of itself. From there, move to the neighboring cells, and rapidly throughout the entire plant. And what I will do, like in the next few slides, is to show you like each of those different sequential steps. So, something to understand, a failure of a virus to go on through each one of those steps, it's a failure of a virus to infect. So maybe it's not clear, but it is not that easy to be a virus. And first step, to enter the cell. What is the first obstacle? The first obstacle of a virus is the cell wall, a barrier that is impenetrable for the virus. And so the plant virus has to find ways, adapt to go through it. As smart as they are, what they do, they just hitchhike. They're hitching a ride from fruit flies, not fruit flies, aphids or white flies. What indeed is happening is that like during feeding, those insects are able to pick up the virus and just release it rapidly inside the cell. And those are like plant-sucking insects, so it's a way for the virus to get in directly inside the plant cells. What is happening once the virus is inside? It's time to make copies, and as you can see, like here, the first thing that the virus will do, it will be to, to disassemble like its shell to release the viral genome, and from there, take over the cell machinery and make copies and copies of its genomes. It is estimated that like a virus is able to make about 200, 1,000, or up to 1,000 viral copies per cell. So let's just imagine, I told you that we are made of 50,000 trillion of cells, and imagine that if in each of the cells a virus is able to make such amount of copies, what can happen? We'll be dropping dead, I guess, by now. But following, once the virus is successfully multiplied, it's time for it to move to the neighboring cell. And this is what we call like cell-to-cell movement. And where the virus is like, what they do, like they will have to use like a tiny channel that are connecting neighboring cells. Like those are little openings. What are those openings? Those are normal openings. We refer as plasma this matter. You don't need to remember that. But those are openings that a cell will use normally like to circulate and distribute like water, mineral, like in a normal way. And the virus will just take advantage of that. Something for you to remember. Yes, viruses are very small, but they are not as small as a molecule. So you can already sense there must be a problem right there. What type of problem? And this is my way of describing it. It's like to try to sneak in a big elephant through a normal sized doorway. What it means is that the virus is way too big to get through those tiny channels. What does it mean? The virus needs to find a way to get through it. And it's how I think that viruses are just amazing. Back again, with 4 genes, they're able to do that. What they do, they're able to destroy those tiny channel to be able to fit in. As illustrated here, if this is the normal size of those tiny channel, the virus is able to enlarge them enough to be able to fit in through and move to the next cells. Talking about the speed of it, you can imagine, I didn't mention it about like when you are transmitted by aphids, this can happen within seconds. But in this case, like the modification of a channel will take about micrometer per hour, so a very slow process. However, once the virus manages to do that, this is it. It can just spread throughout the plants. And it's like, how does the virus spread throughout the plants? It needs to reach the vascular tissue, which is like literally the plant's highway. This is a rapid movement where a virus can move within the plant in centimeters per hour. So to summarize this, the sequential spread of a virus, if we start from the initial viral infection in day 1 from the leaf, you can see where the virus first will move through the roots. From the roots, it goes up to the young shoot, and from there, reaches like the other tissue. 25 days from the day 1 that the plant was inoculated, the plant will literally die. So with that, I wanted to conclude that about this plant virus race, why it is a race. It's not a walk, a promenade of a virus. It's a race because the virus needs to fight also against the host defense response to get through its life cycle. Okay, thank you.
Hang on a second, I'm not used to the Mac. Somehow I have to find down here and say, here we go, I think. I've got to find the bar down there. I'm a PC person, I'm sorry, you're probably gonna, shoot me down. All right, thank you, Dan.